our first biochemistry episode. Today we're talking about enzymes. Enzymes are proteins that are able to accelerate biochemical reactions within our bodies without being consumed by it. Enzymes can be seen as biological catalysts, and as I said, they accelerate chemical reactions. And the most important or an essential aspect of them is that they're not consumed or altered by it. The basic principle is that they are capable of lowering the activation energy, and the activation energy, as the name suggests, is required for a reaction to take place. And this is what you see here. So the two main criteria to call something a catalyst is accelerating a reaction and that it's not altered or consumed by the reaction. So maybe the most intuitive way for lowering the activation energy is via providing another reaction pathway. Here often intermediates are formed. The other two forms of lowering the activation energy is that the substrate itself is destabilized or that the transition state is stabilized. So they kind of complete each other. For organisms, enzymes are essential, as energy resources are often not abundantly available. The whole process usually takes place like this. We've got a substrate which is specifically bound by an enzyme, and this can happen due to its charge, shape or other features like hydrophobic pockets. Hydrophobic means that it doesn't like water. Here are two models. You might have heard or you might have learned the lock and key model. So here it says that the geometry of the enzyme and the substrate are specifically complementary. This model was developed at the end of the 20th century. Maybe not that accurate. The current model is the so-called induced fit model. The active part, so that's the part of the enzyme that interacts with its substrate, this can be reshaped depending on the interaction with its substrate. As soon as the two of them are bound, the actual catalysis takes place and the products can be released. And this is what you see here. As I said before, but it's very important, so I'm going to say it again. The enzyme is not changed during the reaction, so it can catalyze the same chemical reactions over and over again. Metabolic pathways like glycolysis or the citric acid cycle, and we're going to talk about them in another video, they would work too slowly without enzymes. So enzymes are necessary for life, basically. There are more than 5,000 catalyzed reactions which are known nowadays, and they can reduce the time span of a reaction from millions to years to milliseconds, which is incredible. The inhibition of enzymes is, by the way, a common method for drugs. For example, acetylsalicylic acid, which you might better know as aspirin. This inhibits the cyclooxygenases, which reduces signs of inflammation and pain. As we're talking about inhibition, there are two major types, which are competitive and non-competitive. Well, as the name suggests, competitive inhibitors means that they're capable of blocking the active side of the substrate, so the substrate cannot be bound by its enzyme. Non-competitive inhibition, on the contrary, means that it binds somewhere else, so the affinity for the enzyme to its substrate stays the same, but the maximal rate at which an enzyme can bind to its substrate is reduced. The rate of reactions can be mathematically described by the so-called Michaelis-Menten Equation, which is the following. Km corresponds to the concentration of the substrate, where the rate of the reactions is 50% of Vmax, so the maximum velocity of the reaction. So a small Km means that the affinity of the enzyme to its substrate is high. As not a lot of substrate is required for an efficient and speedy reaction. Natural occurring enzymes, by the way, all work more efficiently than man-made. And even though the reason is not fully understood, it's possible that it's due to the specialization of the enzyme for the space within a cell. Enzymes are capable of accelerating reactions by the factor 10 times 6 up to 10 times 9. I hope you liked this episode and see you in the next one.